Hello everyone, my name is Kevin Yang, and I'm currently calling from my home in Michigan. I decided to study this project, determining the genetic factors of nicotine dependency, because I noticed the increasing issue of nicotine usage, especially due to e-cigarettes and the increasing audience of them among younger audiences. So, as I previously said, cigarettes are now being targeted towards younger audiences, and not only are they on a wider range of audiences now, but also they're much more harmful, having much more of the harmful chemicals than even cigarette smoke itself. And through this study, I believe that finding these genetic factors and genes associated with nicotine dependency will help in identifying some sort of function or some drugs that can determine if we can accurately predict smoking status from gene expression alone. So first thing I did in my project was look into past studies. These past studies studied different parts of the body, as well as different tissues and things like that. And they use different techniques as well. So typically they use things such as microarray analysis or RNA sequencing. And one of the common conclusions that they all had was this one particular gene, SERPINA1, which you can see on the right here. Serpina-1 has been found to be associated with the pathogenesis of chronic obstructive pulmonary disease, also known as COPD, which smoking is an obvious contributor to. And such studies rather than focused on the identification rather than driving a method or function, which is what I aim to do. So this is the kind of preliminary analysis I did. So the data I looked at was GSC 171936, and this RNA-seq data generated in the nucleus accumbens. So the nucleus accumbens are a very addiction-relevant part of the brain, which makes it a good place to study for nicotine addiction. And this data set included 223 deceased individuals, 50 current cigarette smokers, 171 non-smokers, and 2 individuals with undetermined smoking status. So I first plotted these two genes I found from previous studies, SERPINA1, which I mentioned before, and NOL4L, which I had not previously mentioned. And plotting these based on log TPM value and smoking status. TPM value means transcripts per million, which is kind of just a genetic expression indicator. And it shows that very clearly mean, the mean value of the control for apologies, mean value for the control of SERPINA1 is a little bit higher than the mean value for the case, which kind of shows that, well, there is a difference between the case smoking and control non-smokers, and it's just that it might be an indicator of smoking status. However, when you look at NOL4L, there's not necessarily a significant mean difference in this data, and that can be visualized by the p-value, which is 0.727528, which is much greater than 0.05, which is usually the cutoff for significance. One possible reason for this, I believe, is that there's a different tissue between the two studies. The study that this gene was found from used lung tissue, and mine used brain tissue. So as for my next analysis, I visualized only the TPM differences would not be enough, and I decided to use full change analysis as well. So any difference in overall expression between the samples or conditions will be normalized out when calculating full change, which is the absolute value of the expression value of the case over the expression value of the control. And this can provide an even more accurate analysis. So here you can see there's a table on the right here is the full change values. And I ordered these descendingly, meaning that the highest ones have the highest full change and theoretically the most significance in the genetic basis. So while the numerous values contain values greater than one, almost none of these seem to be found of previous genes of interest, which is very interesting because things like SERPINA1, which are considered very significant in other studies, should be here, right? And then I believe that's likely due to the outliers because although I use median to calculate this data, it's still skewed by many other factors. So the next thing I did was do a train test split. I wanted to find a way to do a logistic model of this, and I knew that in order to make sure my data wasn't just able to be accurate on my own data, but also on new data, I had to do a train test split. So what this does is that it splits the data. In my case, I split it 80% to the training and then 20% to the testing, meaning that the 80% will be tested onto the 20%. This allows for less false or inaccurate results, which is seen in many other studies. I also just used GLMNet. So this is the function I use for logistic regression. And although it normally would be clear, the correlations will exist based on accuracy and predictions found. This allows it to be even more accurate and show a proper prediction table, which I'll show later. I use GLMNet particularly because it helps me tune the regularization parameter by using cross-validation. In my case, it was five-fold cross-validation, which helps to optimize the model's performance on new unseen data, which is what we did with the train test split. Next thing I did was create a confusion matrix. So this is the thing I mentioned before where it helps show the visualization of the predictions. So as you can see here, the two case values here show there's one true positive for case and 32 true positives for non-smokers, or yeah, for controlled non-smokers. And there were 11 false positive results. And as you can see here, the accuracy is actually pretty decent. For a study like mine, this would be enough, 75%. But what's interesting is that most of the inaccurate results were found through case. So only one right case prediction was made, which shows that maybe the data is actually skewed towards control and that these aren't actually accurate results. So in the study between the relationship between gene expression and smoking status, I believe it's inconclusive but requires further study to determine whether the low accuracy for case patients is due to the gene's insignificance or the influence of the outliers that make it look insignificant. One possible reason for this discrepancy, as I previously mentioned, was the presence of outliers, which cannot be truncated completely using median to calculate full change values. One solution I came up for this, which I'm currently studying, is rather than using GLMNet, I use random forest classifier instead, which would be a better predictor for this dataset. Random forests are robust outliers because they get averaged out by the aggression of the multiple tree output. So for example, if you see on the right here, each little tree branch makes its own decision and finally goes up to one decision as yes, no, yes, no. And then once all those yeses and nos are counted up and averaged together, the final result will be the final decision. So in my case, even if one tree says, no, it's not a smoker, but all the others say it is a smoker because of outliers, then in that case, it will be a smoker, and that'll be a much better result. So my further studies for this is I'm currently looking into a different data set, which is GSC 4419, which contains lung cancer patient data, along with indicated smoking status of them at the time of death. Using the same methods, I am performing the same methods to determine whether genes of interest, typically found in other studies, appear in my own analysis. If the genes such as SERPINA1 appear in my full chain analysis, and the logistic model shows a more interpretable and accurate set of predictions, the other genes found beyond previously found genes will likely also be genes of interest. However, if I find the genes that I found in this study, then it shows that outliers may have affected the results of my current project. And with this greater set of genes, better predictive models for determining smoking status could be developed, including the possibility of determining potential for becoming addicted to nicotine. These are the references I used during this project. Thank you for listening.